Good morning, good afternoon, and good evening, everyone. So welcome to the APSA-NSTDA-KMITL project collaboration on the identification of the SNP markers associated with resistance to pepper yellow leaf curl virus in chili pepper. My name is Kuna, APSA Technical Coordination Manager, and I will be with you today for the whole session. So welcome, everyone. Before we start the session, may I just explain you a little bit about the housekeeping rules. So on the below of your Zoom, you see the chat box. So if you have any technical issues, comments, please put it in the chat box. And if you have any question to the researchers today, please put it in the Q&A box. So remember, question to researcher, Q&A box. Any technical issue, chat box. Participants are not allowed to open the microphone. So if you have any questions, please refer to either Q&A box or chat box. And you can also click like in the Q&A box. So questions that got most like will have more priority to be answered by the researchers. So with that, let me move to the outline of the session today. We will first starting with the proposal presentation for around 20 minutes, and then we'll move to the Q&A session regarding to this collaborative proposal, which we got questions from companies interested in this project, and we have collected all, and the researchers will reply them today. After that, if you have any additional questions, please feel free to ask in the Q&A box. And then I will inform you about the project timeline and action after this Q&A session. Without further delay, may I invite Dr. Viranda Putakram, researcher from the National Omics Center from NSTDA, and also Dr. Pacharapon Suwal from KMITL to present on the proposal. I think for this, Dr. Viranda will be presenting. So please, Dr. Viranda, the floor is yours.
Hi, everyone. I will be presenting on this project. So let me share the screen quickly. Can you see the slide?
Yes, perfect.
Okay, it's full screen now?
Yes,
okay.
It's in the full screen.
Alrighty, okay. So today we'll be presenting on the project that we like to identify SNP markers that are associated with resistant to pepper yellow leaf curl virus in chili pepper. And this will be in collaboration with Dr. Pacharapon from KMITL, who's also here to answer your questions at the very end. So, as most of you are probably familiar with the pepper yellow leaf curl disease, it's caused by virus, and it's a pretty serious disease in a way that it limits the pepper production in most Southeast Asian country. It's pretty bad in Thailand, as well as in Indonesia and Vietnam, in South Asian country as well, like India, Bangladesh, and Pakistan. The virus is transmitted via whitefly insect vectors, and that's why the current management is mostly through insecticide use, which is not very encouraging in a lot of countries. And as we all know, pesticide use, heavy pesticide use is bad for our health. And so we try to figure out a different way, looking for a resistant strain to use instead of the insecticide. The characteristic of defected plant or infected plants is that the leaf will turn yellow and becomes curly, and eventually the plant will stop growing. And if it managed to set fruits at all, the fruit size will be a lot smaller. And in terms of yield loss, you can get anything from 50% to 70% or sometimes the entire field is affected and the yield loss is as high as 100%. And so pretty much everything is wiped out. So this is what it looks like when the chili pepper is infected with the bigamo virus. You can see on the upper left here that the leaf is curly and it's yellowish, and the fruit size is affected. It's also colorized. It turns yellow. It doesn't look very healthy. And obviously you cannot sell these, and so it affects the yield production. There have been a number of resistant sources, genetic sources that have been identified over the past decade or so. And the one we are using is the PSP11 that was obtained from the World Wedge APRDC. There are several sources for each of the capsicum species. However, the QTL have not been identified until very recently. There was a paper that was published a couple of months ago on the QTL that was mapped for the resistant loci. And so what we're going to be presenting next is from the World Vegetable Center, the PSP11 derivatives. And so what we have is what's called PEP6. It's an inbred line that we developed, that Dr. Prashapon developed from PSP11. And this is what it looks like. PEP6, it's, we have shown that it's resistant to the yellow leaf curl virus Thailand version. As you can see here on the left-hand side, this is the resistant plant. And even if you graft the susceptible scion onto it, the resistant plant remain resistant when exposed to the viruses. And on the right-hand side, this is the phenotyping that was done through the use of the insect vector, the white flies. And you can see we alternate the role between susceptible plant and resistant plant. And we confirm that the resistant one were actually exposed to viruses through PCR. So when we try to amplify the viruses from the leaf, we detect it. And so we know that the plant is actually resistant. So what we propose as a method of identification of SNP markers that are linked to the PepYLCV resistance, we have, as I mentioned earlier, we have developed an inbred line PEP6, which have been through multiple rounds of self-fertilizations. But the strange thing about this is that no matter how many self-fertilizations you've gone through, it always segregate resistant and susceptible progeny. And now we have the F6 progeny, both susceptible phenotype and resistant phenotype. So what we propose to do is that we take both of these progenies, which presumably have a pretty much same background, except where the resistant loci are. So we're going to do a whole genome sequencing of susceptible progeny and the resistant progeny that came from the same generation of selfing. And then we will be comparing the genomes of both types to see where the differences are. And so this is what the PEP6 looks like. The fruit is a typical reddish color that's desirable. Okay, so this is what we expect to see from the genome-wide sequencing and SNP identification. So the susceptible progeny and the resistant progeny should be pretty much the same everywhere else in the genome, except the loci where the loci that confer the resistant to yellow leaf curl virus. Another approach that we proposed earlier was to use a commercial line in Thailand called Jindanin backcross to PEP6. Jindanin has all the desirable phenotypes of chili pepper, except that it's not resistant to the virus. And so we'll be integrating the resistant source from PEP6 into Jindanin, and then go through several rounds of backcrosses. And I believe now we are at the third backcross. And so we will take the resistant backcross progeny from the third backcross and compare it to PEP6 parent as well as Jindanin parent. And so what you expect to see is that when you compare it to the susceptible parent, the backcross progeny would be similar to gindanin everywhere except the loci that confer the resistance. Very similar to the approach we took with the PEP6 self-fertilization progeny. And since a number of companies have asked about resistance to the yellow leaf curl Indonesian version of the virus, we propose an alternative line. So in case you're not so interested in the gindanin, which is the commercial line in Thailand, you might be interested in the PEP12, which is resistant to the PepYLCV Indonesian version. However, the reason that we didn't propose to work on PEP12 in the original proposal earlier was because that PEP12 has a somewhat less desirable phenotype for the fruit color, and so the plants themselves, it's got a lot of anthocyanin. The young fruit is somewhat purplish, which is not really the desirable color for chili for the market. And when it's ripened, it's also not bright red color that you usually see in the market. It's like somewhat orange. So this could be a concern, but anyhow, PEP12 is resistant to PepYLCV Indonesian version, and we also have PEP12 self-fertilized, although not as many rounds as PEP6, but we can keep we can continue selfing it until the background is pretty much the same. So the progeny should essentially be inbred, except the resistant loci, which again, the resistant phenotype segregate. So I assume that the genetic basis for the resistant is not straightforward. It's not a simple recessive. It could be dominant, but then the homozygous might be lethal, or it could be there could be some epistasis, very complex genetics basis, which we would not go into. But we were hoping that once we self it enough time, we can compare the resistant and susceptible progeny the same way that we were going to do with PEP6. And so we'll be comparing the genotype of the susceptible progeny with the resistant progeny and try to identify regions in the chromosome that are different. And hopefully those will be the regions that associate with the resistance. And once we have found SNP loci that are associated with the resistance, we will be validating it using the progeny from a PEP6, PEP12 cross. And so we'll be crossing these two. PEP6 is resistant to the Thai version of the virus, and PEP12 is resistant to the Indonesian version. So the F2 progeny could be used for validation of both sets of SNP, both the SNPs that are associated with the resistant to Thai yellow leaf curl virus, and the PEP12 background will be the source of the resistant for yellow leaf curl virus for the Indonesian version. And hopefully that's not too confusing, but if it is, please shoot your questions in the question box, and Dr. Pacharapon and I would be happy to answer the questions now. Thank you.
Thank you very much, Dr. Viranda, for the presentation. So I think for the next, we can move to the Q&A session that we receive from companies. And then I also saw a question in the Q&A box. So in the meantime, if you have any additional question, please feel free to put it in the Q&A box and we'll get back to that. Okay, so let me share the presentation. Okay, so we received a few questions from APSA companies regarding to this project, and may I request Dr. Pacharapon and Dr. Viranda just to lead the session according to the first type of the question about the validation, please.
Okay, so Dr. Prachaphan will be handling questions for the validation.
Okay. Hello, everyone. My name is Pachalapon. For the first question, for the resistant lines, what performance in the fields or artificial inoculation screening. For the PSP 11 and PEP 12, PEP 6 and PEP 12, and yes, we screening in fields for the first time, and after that we screen by grafting and Wi-Fi methods. Yeah, and we confirm that it can be resistant, but the resistance still secret in the F6 populations. And we never tried in South Asia before, but we try to screening with PEP Thailand, PEP Thailand species and Indonesia species. PEP 12 can be resistant to both PEP Thailand and PEP Indonesia. And PEP 6 can be resistant to and are you also resistant? The resistant line in the PEP 6 and PEP 12 we can share with the Thai company and register how to say in the company that we have registered in Thailand, we can share the PEP 6 and PEP 12, but for other country we can share with F2 population with cost between PEP 6 and PEP 12 under MTA documents for research. And the question number 5, if the project duration is of over 3 years. Oh, okay. Maybe in the first year, the partners can use the F2 population for screening in your country or your area first, because of now we have F2 population already between cross of the PEP 6 and PEP 12. You can sign the MTA for F2 population for screen first. Yeah, and sorry, the question number 4. What are you validating strategy for marker response? I think the resistant material from PEP 6 and PEP 12. PEP 12 have broad resistance, means it can be resistant to pepper yellow leaf curl virus disease. But I'm not sure, so you have to try with the F2 population first. Or if you want to use the germplasm, you can request from Berwick from Taiwan. Yeah, because we received the resistant sort from Taiwan also, PSP 11 and PEP 12. So we can give the pedigree names. Yeah. And please go to the next place. Yeah. The value of trade in South Asia, we need to confirm the web in Venus country. Yeah, as I mentioned, you can use the F2 population for try first, and you can know the material can use with your country or not. Okay, for the projects of renewable resistant source, yeah, for confirm the resistant source, so we try to inoculation with grafting and use the Wi-Fi and detect the virus infection in the resistant plant, and so we got the virus infection in the resistant plant is still a healthy plants and can produce the fruits and give the high yields. It's like PEP6. And now we do F3, F2, and F3 population. We screen in F2 and F3 population. They can transfer the resistant gene from the resistant to the susceptible one. So we have the information for the inheritance of the resistant genes, right? So we can confirm they are the resistant parent is really resistant ones. Number 4 was it the strain race of okay. For these projects, we use PepYLCV isolate. And if the country, if the some company want to use Indonesia isolate, so we will add this for screening too. Yeah, and number 5, okay, we can share the F2 populations. Yeah. How long resistant includes to the SNP? The resistance of equals to, yeah. Maybe cover.
I think for number 6, the resistance source will not be included as deliverable. Only the SNP markers will be deliverable, but you can obtain the resistance source directly from AVRDC and integrate that into your whichever background you're using. Unless, of course, the company is in Thailand. So because we have issues like exporting and importing stuff across the border.
For number 8, the segregation of the resistance from PEP6 factor is wrong. Confirmed by number 6.
I think Dr. Pracharapan is pretty sure that it's genetically inheritable, the trait. I mean, I think the question is asking whether or not it's genetically, the resistance is genetic or environmental.
Oh, it's genetics, and I think it's controlled by genetics and also environment, because of there are polygenic genes control. Maybe if but the main is from genetics control. Number 9, the confirmed resistance by single recessive genes. It's not single recessive gene, right?
It's not recessive for sure, because we cannot get an inbred line that remain resistant. And so my personal guess is that it's dominant and the homozygous is lethal, or something more complicated than that.
Yeah. Number 10, I'm going to consider a broad spectrum resistance. In F2 population of PEP6 crossed with PEP12. So we will test the QTL associated with resistance, right? And after we got the SNP marker, right? Yeah.
I think the main role the companies would be providing funding and will take care of all the experimental design and carrying out the experiment and final analysis to deliver the location of the SNP markers that are linked to the resistance.
Yes, for APSA companies who join the project, they will also receive the report that the scientists will keep updating for that as well.
All right, as for the questions on markers, number one, will there be multiple SNPs? Yes, of course, there will be many SNPs. Hopefully there'll be clusters in one or two or three, depending on the number of loci that are responsible for the resistance. So we expect to find several QTLs, and within each peak, there will be several SNPs. And which SNPs has the highest lot score, we will design the assay based on those SNPs. Second question, can we use the resolved SNP marker for genomic breeding pepper in Indonesia? If we use PEP12, yes. If we use PEP6, no. Because only PEP12 is resistant to the Indonesian version of the virus. Number three, whether a company can receive research results such as resistant genetic resource. Dr. Pacharapon already addressed the genetic material question earlier, but you guys can get the marker location as well as sequencing data, which is also in number 5, whole genome sequencing data and comparative genomic data be accessible. Yes, if you participate in the program, you will receive the raw sequencing data that came out of the project. Field data and phenotypic data will also be shared with participating company. And yes, number 7, genotypic data will also be shared with the participating company. Yes, you will have access to the sequence data and phenotype data and the analysis, as well as the final report. So, there are some questions in the Q&A. I'm not sure if
Yes, so there are a few questions in the Q&A box. And I think we can also go by, nobody's clicked like in each question yet. So maybe, may I request the scientist, when you select the question, please read the question first before you answer the question.
Okay, I'm not sure how.
I can start with the first one from Mr. Davinder Sharma. In India, we have problem of chili leaf curl viruses. How this project will help to breed resistant varieties in India?
Dr. Pacharapon, do you know if the Indian version is similar to either Thai or Indonesia version?
Actually, for the Bigomovirus complex disease, so we don't know the resistance sort that we have can resistant to chili leaf curl virus or not. But so for the resistance that we have, we don't know. we can they can resistant to the chili leaf curl virus or not. I cannot answer it because they have, but I have some research. They report the resistant variety to chili leaf curl virus. Maybe can find some resistant later, but we don't know. We never trace before for chili leaf curl virus disease.
Okay, I think we've got a few more questions from Indian breeders as well. So I'll just go by the time first that they ask question. So next one is PEP6 and PEP12 both are carrying recessive gene for PepYLCV?
I don't think so. Well, or there seems to be multiple loci. One of them may be recessive, but I don't think all of them is recessive.
Yeah. We have the information about F2 segregation between PEP6 and PEP12. In the F2 generation, they have segregate one resistant and 15 susceptible in F2 populations. But in the resistant line, PEP6 and PEP12, they have segregate the ratio one resistant and one susceptible all the time, maybe 3 times already, but we is quite not sure they have all recessive or not. Maybe have some loci dominant genes controlling also.
Okay, I think there are more questions coming. So once again, attendees, if you click like questions, you can also have the priority to be answered for those questions. And for questions that cannot be answered within this session because of the time limitation, the scientists will be receiving questions and we will share to you after the session. Okay. Next, the elite line developed from World Wedge line also should be tested in major markets to know the value of the S&Ps before closing the project. Testing for LCV is based on MSI or white fly mediated. If white fly mediated, what race of white fly? And Dr. Sitaram also said that World Wedge is clear it's not effective in South Asia, but may work in Southeast Asia.
แมลงหวีขาวนี่เรายังไม่ได้ทดสอบเนาะ
Oh. What kind of fly did we use?
Major market
Do you know what race or what race of fly that you use for phenotyping? Dr. Petrova? No?
I don't know. So
Maybe this question can be explained later, and we'll share this the answer later for this question. Okay.
Yeah.
Yes. So the question will be shared to the scientists later on. Okay. Next. Will pathology screening be used together with S&P marker screening for the validation studies, PEP6 Jindanin BC population and PEP6 crossed with PEP12 population in order to confirm the efficacy of the S&P markers correlating with true resistance?
Yes, I think we'll do phenotyping of the F2 progeny that we use for validation. And so we'll know which progeny is resistant and which progeny is acceptable. And we also will take a leaf, extract the DNA and do a SNP genotyping on those progeny. And then we'll compare the ability of the SNP marker to predict the resistant phenotype that way. So yes, we'll do both phenotyping and genotyping and compare the data.
Okay, so next question is from Dr. Sitaram. The materials on which we develop S&Ps must be tested in India before we know its utility for this market, which is key for funding for Indian companies. So how do you plan for that? And if you have any comments, I think there are a few companies interested in this project. Yeah, so they would like to know more information.
Yeah, so I guess you can obtain the PSP11 from WorldVest, but I saw some comment earlier that it doesn't work in South Asia. I'm not sure about PEP12 original strain. Do you know what's called what it's called by WorldVest Center? The PEP12 original strain? Maybe the PEP12 would work in India because it's resistant to both Thai and Indonesian variant. Maybe it's resistant to the Indian variant of the virus too. But maybe Dr. Pacharapon can look up the original strain or the original accession obtained from World Vegetable Center for what we call PEP12.
It's number 9853-123. It's number.
Maybe you could type it into
In the chat box.
Chat box.
Yes.
Okay.
Yeah.
Okay.
Yeah, there's a comment that we may get the opinion of paper breeder from WorldVeg about this as well.
You see the answer.
Yes. Okay. So, yes, shall we move to the next question? Will you be able to determine the percentage, explain phenotypic variance for each marker related to PepYLCV resistant? And will this information be important to know which markers are associated with major and minor resistant genes QTLS?
That's a very good question. But the way we are doing it, I'm afraid we might not be able to determine the percentage explained because we're not doing a population base. We're only selecting inbred lines that just happen to segregate for this trait and comparing the genomes of both, looking for the areas that are different. And so we're only doing like 2 to 3 individuals, like 3 susceptible versus 3 resistant might not be enough to generate a linkage map and do the traditional QTL analysis. But when we do the analysis of the single marker, the lot score or the score of how well they link to the trait may reflect the percentage explained, the percentage that loci explain the phenotypic variance. Because usually, even though there's no direct link between lot score and the percent PVE, the marker that have very high lot score usually are the major QTL. And so we can make an educated guess from the lot score. But in a traditional sense, we won't be able to get the percentage explained phenotypic variance using this method. Yeah.
Right. Thank you very much. So the next question is, the LCV testing is based on MSI or whitefly base. If whitefly, what biotype will you use? For this, yeah, I think I will request the researchers to get back to you later on for this question. Right, and the next question is from Mr. Sunehiro Yagoshi. Will deliverables include resistant lines which you will make in this project? I think you have already explained in the presentation, but maybe you can repeat once again.
So if your company is located in Thailand, you will have access to the material. But if you located outside of Thailand, the deliverable will only be the SNP marker location along with the sequencing data and the analysis information, because we cannot export the genetic material outside the country. So yeah, depending on where the company is located. If you're located in Thailand, if you have a subsidiary located in Thailand, then you will have access to some of the materials in from this project. But if not, you can request the resistant source from the World Vegetable Center. The PEP6 is PSP11, and the PEP12 is some number that we can provide that information later. I don't remember the session number, but both of them are available from the World Vegetable Center.
And as well, you can use we can share the F2 populations.
Oh, and the but the F2 progeny between PEP6 and PEP12 can be shared. Yes.
Thank you very much for the clarification once again. Okay, so next, I think this one is more a comment. Indian bergamots are more virulent and mixed as compared to Thailand and Indonesia. Yeah. Just very
Wow.
right. And the next question is from Sosenti. When is the decision on using either PEP6 or PEP12 will be made?
Actually, the original proposal would be PEP6 and PEP6 Jindanin backcross. And now we're proposing to replace the Jindanin PEP6 backcross with PEP12 cell fertilization progeny. And so if companies are more interested in PEP12 rather than Jindanin, both PEP6 and PEP12 will be in the same project. And so we'll be doing both alongside each other, and we'll just drop out the Jindanin unless there are companies in Thailand interested in Jindanin, because Jindanin is a commercial background in Thailand. So yeah, so I don't know what the makeup of the participating company would be like. Yeah, but we can do both PEP6 and PEP12 if nobody's interested in the Jindanin.
Okay, right. Moving to another question. If you are going to use backcross population for SNP discovery through developing process, how are you making sure that you will not miss that all the majors and minor QTLS, as there is no genetic local information already about the resistance?
Oh, okay. So while we're doing the backcross, we select for a very resistant progeny after each backcross. And so by assuming that the resistant progeny has all the major and minor QTLs, after each backcross generation, all the necessary QTLs were passed on to the progeny that show resistant phenotype. Yeah. So usually Dr. Pacharapon will select the most resistant progeny to be the parent of the next backcross. And I think that pretty much guarantee that at very least the major QTLs are there. Yeah.
And before we backcross, so we will sell for F2 generation for select the resistant one and use the resistant one backcross to the Jindanins or the female parent. Yeah. And so we will make sure the resistant before backcrossing. So I guess we can get the resistant genes.
Thank you so much for the clarification. So we are almost done. The next question is from Mr. Agavi Kandito. Based on several researches conducted in Indonesia and the nature of bergamot virus itself, there are commonly found mixed infection between PepYLCV and MYMIV, PepYLCKAV, etc. So is PEP12 or PEP6 resistant to those viruses?
Mhm. For PEP Indonesia, yes, PEP12 can be resistant, but for I don't know MYMIV what's the virus, but for TY Kanchanaburi virus, so yeah, it can be resistant in the field that we screened maybe 3 years ago. They can resistant, but we need to but we don't confirm with the PEP6 and PEP12. Yes. Mhm. But before this PEP6, they have resistance to PYLC Kanchanaburi virus disease.
Right. Thank you so much. Okay. Next is, could you please let us know the fruit length and pungency level of PEP6 and PEP12?
PEP6, so okay, let me see the information. PEP6 is 5 to 7 centimeter, and PEP12 is 4 to 6 centimeter. And the pungency of PEP6, I how to say ยังไม่ได้ทดสอบ
Oh, we haven't tested for the pungency.
Yeah, that's okay. We got a very good questions actually. Right, and we also got a few comments, valuable comments on behalf of South Asian companies about this project as well. So Dr. Sitaram also commented that instead of very strong resistance to Thailand or India strains, a medium tolerant, stable with wide spectrum resistance would be good. So just for your consideration about that. And yeah, the Indian companies also have to consider about the value of this project, and what works in Thailand and Indonesia may not always work in India. So just for your consideration for that as well. Right. The next question is from Mr. Prakash. It's the complex viruses that hampers production of pepper cultivation in India. If this project is focused only on PepYLCV, then how about resistant to other viruses?
I think for find the marker, we should be use how to say find some specific genes to the major disease one first. But yeah, it's virus is very complex. Yes, how to say.
Yeah, hopefully there will be more research for other viruses in the future.
Yeah.
So thank you for your comment, Mr. Prakash. And the last question as of now from Josna. Would you also be studying candidate genes conferring resistance? If yes, will you be providing that information also to participants?
Oh, yes, definitely. So once we get the SNP markers, we will compare it to the genomic region of the published genome and see if there are any putative resistant gene in the vicinity of the markers. And if we find something interesting, we could definitely share that information. But anyhow, you would know the location of the SNP markers. And so if you have some information from other sources that that chromosome has resistant gene, you can compare and see how closely they are between the markers and your gene of interest. But yes, we will provide the putative gene within the SNP marker location that we identify.
Thank you so much. So we are done for the question from the Q&A box and also the question in the chat box. We have also copied that. So next is to share with you the timeline of this project. Okay. Right. So according to the project timeline, the companies can consider and get back to me by the end of April or April 30th. After that, I will send emails to all companies asking about the confirmation, and then we can inform the final numbers of participating companies so that we know the amount of the project to be joined by each company. So after that, in the middle of May until end of May, it will be the process of the contract signing, invoice sent, and fund transfer, and the project will start by the timeline on 1st of June. So that's the timeline, and after this, we can also share the video link to your colleagues in case some of your colleagues did not have a chance to join the meeting. As of now, we will share the video to you, and you can consider get back to us by the end of April. So thank you very much for today, and we wish you have a good day. Okay. Thank you, Dr. Viranda, and thank you very much, Dr. Pacharapon.
Thank you.